Hello, friends. We are now starting part 2, the 2018 Biology State Test. Here's our first question, number 13. The following questions has two parts. First, answer part A, then answer part B. Which statement describes crossing over as it occurs in meiosis? Remember, crossing over is where the homologous chromosomes in prophase 1 of meiosis 1 touch each other and exchange genetic information. Okay, so which statement describes crossing over? Is it A, the allele of a gene separate? No, that takes place in anaphase. B, spindle fibers pull apart sister chromatids. Again, in part of cell division, anaphase. C, genes move to opposite ends of the chromosome. Well, this is how translocation occurs. But D is the homologous chromosome exchange segments of DNA. Just like I was telling you, homologous chromosomes. Dad has chromosomes, okay, that have certain genes on it. Mom has a chromosome identical to that. It has certain genes on it, the same certain genes. And when they touch each other, they exchange DNA. And that's how come in sexual reproduction, we get different offsprings. Part B, which do science use to confirm that crossing over occurs? Well, this is where we use the process of elimination. A, cloning. Well, that's just duplicating things, right? Making things making genetically identical individuals. Let's go to C, gel electrophoresis. Well, you know, that's what we use to separate DNA fragments and compare to other DNA. Genetic engineering. Well, that's where we used a set of technologies used to change the genetic makeup of a cell, including the transfer of genes within and across species boundaries. So in other words, we're putting genes in one species into another. Then we have dihybrid crosses, and this is the correct answer, right? Sex crosses involve two non-linked traits provide confirmation that alleles located on the same chromosome were inherited independently of one another. So when we did a dihybrid cross of the dog, you saw where we had the brown dog, the black dog, and the yellow dog. From doing a dihybrid cross like that, we can determine if crossing over has occurred. Number 14, which molecule was coded for by the longest piece of DNA, assuming that no post-transcriptional RNA processing or splicing has occurred? Well, let's look at B, a polysaccharide with 60 sugars. What does sugars have to do? It has nothing to do with DNA or RNA, right? C, a messenger RNA molecule with 60 codons. Well, we know it takes 3 bases to make one codon. Remember, a codon's a code. And that would be 60. So we'd have 60 bases. A messenger RNA with 45 nucleotides. Each nucleotide only has 1 base. So that means we would have 45 bases. So far, C is in the lead with 60 bases. But if we look at A, a protein with 25 amino acids, it takes 3 bases to make 1 amino acid. So in this case, we would have at least 75 amino acids, at least. Okay? So the answer would be A. An island An island frog population is made up of 2 color phenotypes, green and brown, caused by different coloration alleles. The frequency of coloration alleles is shown in the original population box in the diagram. Because of changes in the environment, green frogs become more visible to predators and brown frogs are better camouflaged. A, place color symbols into the new population box to predict the frequency of frog coloration alleles due to changes in the environment. B, select the evolutionary mechanism that would cause the change in allele frequency. There may be more than one answer. Okay, so here is our chart, okay, or diagram. the frog allele changes. So we're told in the instructions that the birds are eating the green frogs because they're more visible. So we need to add more brown, right? More brown into our new population, right? So we only have 2 here, but if we have 3, okay, and let's put 4 here. See, we have total 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So we have 4, and we have 1, 2, 3, 4, 5, 6, 7, 8. We have 8 green ones. But if we put 6 green ones over here, 1, 2, 3, 4, 5, 6. Well, we have increased the brown population, and we have decreased the green population. So we have 1, 2, 3, 4, 5, 6, 7, 8 over here, 10 over here. 2 of them are brown. Over here we have 1, 2, 3, 4, 5, 6, 7, 8, 9. We have 10 over here, and 4 of them are brown. So our allele frequency has changed. That's evolution. Now, what caused this evolution? Okay, remember, it's because the birds were eating the green-colored frogs. So that founder effect, natural selection, or sexual selection. It is natural selection, okay? Remember, the founder effect is where some group goes and leaves the other group, right? And sexual selection is what you prefer sexually. So this is a good answer. This is the answer for number 15 on the 2018 test. Question labeled 17. Park rangers track the size of a stable deer population in a protected forest reserve. An area of neighboring land is added to the protected forest preserve. Park rangers plan a survey over a 10-year period, B on the graph, to measure how the deer population changes as a result of territory added to the preserve. They set up the graph as shown. So we have the number of deer, and we have time at the bottom. The dark line is the deer population. The dotted line is the end of the survey period. And the red line is when the new territory was acquired. So let's select the boxes to predict the population and carrying capacity. Well, we expanded the territory, so I would say our deer population would increase. If our deer population did increase, okay, then our carrying capacity of habitat during time B would increase. And then during the carrying capacity of the habitat during time C would be at an equilibrium. This will be our last question of this part 2 for 2018 biology state questions. And it is number 18. A student studying feather color inheritance in a certain species of chicken observes that chickens have black homozygous, blue heterozygous, or white homozygous feathers. The student hypothesizes that this feather color trait follows the incomplete dominance inheritance pattern. Now, which table would support the student's hypothesis? When we were talking about a single gene trait, we said if it was dominant recessive, you could only have 2 phenotypes. However, if it was a single gene trait and it could be incomplete dominance or codominance, you would have 3 phenotypes. So what we need to look at, let's not look at the first one, let's look at the second one. We have parent 1 white, parent 2 black, the offspring black, just 2 phenotypes. We go to C, parent 1 white, parent 2 white, offspring black, again, only 2 phenotypes. D, parent 1 blue, parent 2 white, the offspring blue. We still only have 2 phenotypes. But when we go to the first one, back to the first one, parent 1 has black feathers, parent 2 has white feathers, and the offspring has blue feathers, which is different from the white or black. So we have 3 phenotypes. So this would be the correct answer. Okay, that ends our session for today. Thank you.